All right, so this will be the second part of neurophysiology. The first part, we got our neurons stimulated. We started our action potential, and we went through all the gory details of the action potential and the refractory periods. So here we're going to look how the action potential is actually brought down the axon, and then we're going to look at what happens at the synapse, the chemical synapse, where those axon terminals go down and you're going to get communication to the other cells. Okay, so that will be this part of neurophysiology. All right, so how an action potential actually gets moved down the axon, because if you remember, the action potential doesn't lose strength. If you measure at the beginning by the axon HELOC, the action potential is this height. When you measure all the way down at the end, the action potential is the same height. It doesn't lose amplitude as it moves down. Okay, and that is because we're going to re-up the signal basically every stretch of the way on the axon. So this movement of the action potential down the axon is going to be called propagation. Okay, so that's propagation. And how does this take place? Okay, propagation of the action potential. It is unidirectional. So we don't want the action potential going from the axon HELOC down to the axon terminal, not in the opposite direction. Okay, so let's look at this propagation of the action potential. Here's our sodium channels. Okay, and this is where knowing what your absolute refractory period is going to come. Remember, when those sodium channels are inactivated, right, the inactivation gate is in play, similar to this orange one here, that channel cannot open, right? So let's just say this region here is where the axon HELOC is. Okay, this is going to be our first stretch of sodium channels. Okay, and the opening, so we hit threshold, that causes that rapid opening of the sodium channel. Okay, and we get influx of sodium. That's going to bring the membrane potential more positive, depolarize the membrane potential. Okay, that depolarization caused by this opening of these channels, right? It's one channel here, but just think of them as a stretch of channels on the membrane. And the opening of these channels will cause the opening of the ones further down the axon. All right, and so now this channel is open. The key is, if you remember our absolute refractory period, is when the channels are inactive, they cannot open. So when this channel, because this channel is cued by the opening of this channel, when this channel is opening, this channel has already gone inactive. Okay, this channel cannot open. Now we get the rush of sodium. Boom, this one will become inactive. But when the sodium's coming in here, that's going to bring the membrane potential up, and it's going to depolarize it enough to open up these channels, because these are not inactive, but it will not open the ones behind it because those are inactive while this channel is opening. Okay, so that keeps us going in one direction. That's the propagation. This propagation of this channel causes this channel to open. Opening of this channel causes this channel to open. Okay, but the key is the channels behind are inactive, so therefore the signal can only go in one direction. Okay, so that is that there. We're going to look at what affects the speed of this propagation of an action potential. Okay, we're going to look at two factors. We're going to look at myelination, right, and diameter of the axon. And what we're going to see with myelination, we get an increase in speed. It is much faster. And we'll see why. As you can see, if there is no myelin, all that membrane is exposed, and so we have to re-up the channel or re-up the signal every stretch of that axon. Okay. When we have insulation, which is our myelin, we won't have to do that. Okay, the other is diameter. When the diameter gets bigger, there's less resistance on the current, and therefore you can get faster movement of the ions and you can get quicker rate of the action potential. Okay, the other is temperature. When temperature increases, of course, we're going to move faster and therefore increase the rate. We'll focus on myelination and diameter here. Okay, so we already diameter is one of those, and then myelination. You can see this is the early studies on neurons were done on the giant squid axon. You can actually physically with the eye see this is one neuron. You can see it is giant, so you can imagine how fast this can move. If you've ever seen a squid, boom, they move very quick when they react. And that's because they have this large giant squid axon that gives a very fast rate of conduction. Okay, so that's diameter, and the other is going to be that myelination. Okay, so myelination. Now you can see with the myelin, this is the Schwann cells here, and those Schwann cells produce that myelin. It's like a fatty wrap around. It's basically the plasma membrane just wrapping around multiple times in these concentric circles. And if you know, fat is a good insulator, and so if you remember our graded potentials here, there's no myelin, so they could leak out. But in these axons where there's myelin, there is no leaking of the current, and therefore the current, we can open up channels here, they can skip, boom, down here, and that'll give us enough depolarization here to open. So basically we're skipping. In the other slide we saw, we had to open channels every stretch of the way. Here we can open channels, boom, skip, open channels, boom, skip. All right, so in these axons, you're only going to find those voltage-gated sodium and potassium channels in these gaps here called the nodes of Ranvier. Okay, and this skipping, this what we call saltatory conduction. Saltatory conduction is a conduction that takes place down the myelin. That's the skipping, or myelinated axon. That's the skipping to the nodes. All right, saltatory conduction. If there is no myelin, then it's called continuous conduction because we have to continually open up channels along the way. Okay. So here's a look of that saltatory conduction. Okay. Here's our myelin. We've had depolarization take place down there. Because the myelin's here, we don't lose the current. It can move down here, and that makes it so there is enough depolarization when we reach the next node. There's enough depolarization to open up these channels. Opening in these channels, we can skip down here, boom, opening there. So we don't have to open all down the way. So this makes this a much quicker rate of depolarization, quicker rate of action potential. And you see the nodes. Where do we find the sodium channels? Just in those nodes of Ranvier. So this myelination is great. It allows us to speed the conduction. Okay? But there are diseases that may affect the myelinated neurons, can attack. There are certain autoimmune disease in which the myelin, the immune system, attacks this myelin and destroys it. The problem is it's great if it's here because we can skip and we can get this really fast rate of conduction, but the axon is not making channels. There's no channels embedded underneath that myelin. So when this myelin gets destroyed, you can see, boom, opening the channel. Remember that myelin prevented the leakage of current. But now if there is leakage of current, by the time we reach to the next node Ranvier where we find those sodium channels, we may not be at threshold, and therefore that won't open the channels, and therefore the communication of that action potential going down that neuron is going to be stopped. Okay, several of those Z's, probably have heard of multiple sclerosis, Guillain-Barré syndrome. This is where there is a loss of myelin. Okay, it's going to slow the conduction or even prevent it. All right, so let's look at frequency and action potentials. How fast a neuron sends action potentials, how frequent they do. Because if you remember, the action potential is a all or none phenomenon. It is going all the way or not at all. Okay, you can see here is a graded potential. This is reached well over threshold. You can see it's quite over threshold. Okay. Remember the action potentials. Each of these tick ups is an action potential. You can see the height are the same. So the amplitude of the action potential, it goes all the way or none. Okay, all the way or none. But the height is the same. So all or none. The key is that this is a fairly strong stimulus, and so you're going to look at the frequency of action potentials. Okay, decently frequent. Boom, boom, boom. Okay. A little bit of neurotransmitter. We'll talk about the release of this in a second. Okay. Here is the graded potential. This one is much larger. We're up to basically 0. Okay. We've gone up basically 70 millivolts. This is a very strong stimulus, very strong excitatory graded potential. And what do you see the effect is on the action potential? Again, look, the amplitude of those individual action potentials is the same height. It's all or none. But the frequency compared to this smaller stimulus, this stronger stimulus creates a faster rate of frequency of action potentials, more boom, 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 the frequency, and therefore that will result in increased neurotransmitter release, which will also increase the signaling. Okay, so stimulus strength, the graded potentials, how are they coded? They're coded by the frequency of action potentials. My sensory neurons respond, I have touch receptors. Okay, touch receptors respond to touch, and when I touch, it creates a graded potential. I can touch very softly, all right, and I would have a small amount of, or slower frequency of action potential. Now, if I press down that's on that same area, those same touch receptors are getting a stronger signal, a stronger stimulus, and therefore higher frequency of signaling will take place in those neurons that are associated with those touch receptors. Okay, so again, stimulus is coded by how fast the action potentials are being sent, their frequency. All right, so once we get the action potential down, we've propagated it all the way down to those axon terminals. Okay, those little terminals that branch off at the end. You can have thousands of these branches off an axon, then it can communicate to many, many cells. Okay, and that is where we're going to get our cell-to-cell communication. That is how the neuron is going to talk to the cells that it's talking to. But again, we got communication, cell signaling. We got to have a signaling molecule. In this case, what's secreted by the neurons are neurotransmitters. The cell in which it's talking to has to have a receptor. Okay? And then the connection between the neurons and their targets. We will see there are 2 types of synapses, electrical synapses. We'll just touch on, but what we're going to focus mainly on are going to be the chemical synapses. Okay, synapses are little space between, chemical synapses are little spaces between the, say this is the neurons and this is the target cell. Okay. Or you can think of the neuron as the presynaptic cell, and on the other end is the membrane of the postsynaptic cell. Could be a neuron, could be many different types of tissue. Okay. All right, so let's just look at the difference between a chemical and electrical synapse. Like I said, I'll just basically touch on electrical synapses here. These are the fewer of the synapses. They do exist in the nervous system, but they are fewer than the chemical synapse, which we'll look at here. Let's first take a look at the electrical synapse. And you see in the electrical synapse, here's one cell coming in. There's a synapse. In the synapse, there's actually a physical connection. These two cells are tethered together by these connexin proteins. These connexin proteins make up what we call gap junctions. Right? And the gap junctions are essentially little tunnels between the cytoplasms of these two cells. So if I have, say, depolarization here, that depolarization, that ion movement can move on to this cell. And it can go in both directions. All right, whoops, that way. Okay, if I had depolarization here, it can move down into this cell. Okay, so there is an actual physical connection through those gap junctions. All right, the ones we're going to be focusing on and the more prevalent in the nervous system are the chemical synapses. All right, you can see what makes up a chemical synapse. We have a presynaptic neuron, the presynaptic terminal, that's the axon terminal. Okay, but you can see there is a synapse. There is this little space. There is no physical connection between this cell and this cell. All right, and then there is the postsynaptic cell with its membrane. Okay, and so the presynaptic terminal is going to have the neurotransmitters that's going to be released, and then the postsynaptic membrane of the target cell is going to have the receptor for it, so we can get chemical signaling. So what we're going to spend the rest of the time is how this signaling takes place, and look at one instance that will occur with this signaling. All right, so the chemical synapse, that presynaptic neuron has that chemical messenger, has that neurotransmitter, and that is what the function of the action potential is. As the action potential comes down, the action potential is going to be the cue for release of this neurotransmitter. Okay, if there's no action potential, we're not releasing neurotransmitter and we're not getting communication to the target tissue. Okay, but when the action potential comes down, that cues the release of the neurotransmitter, and then we can talk to the target tissue. Okay, you can see the neurotransmitters, like any chemical signaling, that postsynaptic cell has to have receptors at the membrane there. So let's look at the steps involved in the synaptic transmission. Okay. Release. So an action potential is traveling down. Action potential is coming down. We have a new set of voltage-gated channels down here. When an action potential reaches the terminal, there are these voltage-gated calcium channels. Okay, so action potential, we'll see it here, 1 coming down to the terminal. That cues to the voltage-gated calcium channels. And calcium is high outside compared to inside the cell, and therefore calcium is going to rush in. Okay, calcium comes rushing in. All right. Little calcium, these vesicles with the neurotransmitters are just sitting here at the terminal. The calcium is going to cue what we call these snare proteins and that's going to cause docking of the vesicle. Docking of the vesicle, you can see these little snare proteins, little blue ones, and then we're going to get exocytosis of the neurotransmitter. Right, so action potential cues calcium channels open. The calcium cues the snare proteins to cause docking of the vesicles and exocytosis of the neurotransmitter. Now the neurotransmitter is in the synapse. It's going to diffuse across to the postsynaptic cell, and that is where the postsynaptic cell has the receptor, and which is going to bind, depending on what receptor here, it depends on what response is going to take place in that cell. Right after the signal is done, we don't want the signal to stay. Say this was a motor neuron talking to your muscle. You don't want your muscle. Most times, you don't want your muscles to stay contracted completely. You want them to contract and relax. So to get them to relax, we got to take away this signal. All right. In some ways, is we can reuptake them into the presynaptic neuron. There, if it was acetylcholine, there's an enzyme here that can break down. Okay, and so we get signaling. Action potential comes down, cues the opening of the calcium channels. Okay, calcium cues the docking and release of neurotransmitter, the exocytosis, binding, diffusion, and then binding to the receptor. Right, and then finally we have to get rid of the signal, and there'll be multiple ways to do that.